Hello, I'm Jayanthi Grebin, Senior Business Development Manager for Cell and Gene Therapy for CPC. I'm happy to be here presenting at the meeting on the Med, and I would have rather met you in person in Barcelona, but this will have to do till we're all safe. Before we get into the discussion of impact of closed system cell and gene therapy in scalability, let me take a step back and talk about CPC and who we are. CPC is a leading designer and manufacturer of single-use connected technology for biopharm and cell and gene therapy. We are well known for our aseptic quick line, for genderless connectors, for closed system processes. We have over 900 employees in our new global headquarters in Roseville, Minnesota. We have 20 over years dedicated to the biopharm and cell and gene therapy in business. Our products are manufactured in ISO class 7 clean rooms, and they are made right here in the United States and distributed worldwide. We are both ISO 13485 and 9001 certified. That's a little bit of a background about CPC. If you are in the Midwest in US, come visit us. We would more than welcome you. Now, let's switch gears and talk about the exciting opportunities ahead in cell and gene therapy. As we all are familiar with the ARM report, the cell and gene therapy business is growing globally, with US and Asia Pacific leading the way. The Q4 report from ARM stated that there is 1,085 regenerative medicine and advanced therapy developers active globally. Let's take a moment to think this is the next growth market for investment and therapy developers, and how do they need to succeed to save lives? How can we effectively be proactive to bring these therapies quickly and safely to patients in a short time? The molecules are there, the investment is there. We need to work on the method. With the growth comes opportunity and pressure to produce. There are over 1,000 clinical trials globally, with 200 of INDs filed annually through 2025. What does this say? That there's going to be over 20 cell and gene therapy products approved a year through 2025. We are not short on approvals, with the latest being Jayanthi by Bristol Myers Squibb acquiring an FDA approval for CAR T treatment for the treatment of refractory large B-cell lymphoma. As you can see, the number of clinical trials and the anticipated new drug application and cell and gene therapy approvals expected by FDA are large and growing. Research suggests that by 2030, up to 60 new cell and gene therapies could be launched, treating on upwards of 350 patients. What it leads is there will be more smaller cell and gene companies looking for outsourcing. Their manufacturing. All this demand will funnel through a relatively small number of CDMOs and CMOs, which are at or even beyond capacity. Here lies the bottleneck, where capacity overload to sustain this growth. And this capacity has already been tried with the manufacturing of vaccines for COVID demands. So as we have stated earlier, there is definitely scalability challenges with cell and gene therapy. Some have been identified here on the slide. The lack of consistent process and manual processes creates a barrier towards automation, especially in autologous process, to speed up the therapy cycle. Not planning ahead in an investigation for scaling the process increases the cost and time to therapy manufacturing. Working with compressed timelines towards commercialization with life-saving therapies that have fast-track designation has constantly burdened the therapy providers. Cost of labor are expensive, and lack of trained personnel and training operators have been challenges this industry is constantly facing. Working with CMOs and CDMOs and the availability to try to put your therapy in their work stream while competing with other therapy providers. How can this process be cleaner and tech transfer work smoothly? We also have the physical challenges, and limited space reduces the flexibility to make manufacturing efficient and accelerates the need for in-house manufacturing to incorporate closed system and scalable practices. It is effective to be agile in manufacturing with the right partner. Scalability and closed systems are important factor in developing cell and gene therapy. Patients need to have product commercially and viable products available and prepared to get therapy quicker. The value of closed systems has been implanted in the beginning of the process to increase speed with reproducible process. Protect the process, and the employees need to be the #1 priority. Really consider the value of closed system process. Closed system process also have options, and single-use technology adoption provides many well-documented benefits for commercial operation. Some of the things that we have documented here is cost, reducing manufacturing costs by elimination of cleaning and sterilization steps, speed, time and labor, savings during setup and between operational cycles, sterility, elimination of cross-contamination between batches and between operations. The advancement in manufacturing, automation, and closed manufacturing systems are beginning to simplify the manufacturing footprint. The strategy, bringing this treatment to site and facilitating and decentralizing of the operating model. Those are things that we need to consider. The decentralized model will always enable clinical sponsors to provide cell and tissue processing as a spoke to the manufacturing footprint in some of the markets, including what we have seen in the US, Europe, and China. There's an increasing trend in the deployment of closed systems, automation, and scalable for cell manufacturing procedures from starting material to the final cell product. One of the things that we have seen is the thought of reducing biosafety cabinets and downgrading the requirement for space that can effectively help with time and manufacturing of the therapies. The closed system helps eliminate open processing steps, reduces operator transfer between multiple pieces of equipment. In personalized therapy, reducing the risk of contamination from one process to another reduces the chance for product loss that is already experiencing low yields. This opens up the avenue for multiple and flexible manufacturing of different therapies in the same space quickly, cleanly, and efficiently. So the key is to think early. Think early during the preclinical and clinical stages how to scale and close systems from lab to GMP. As I have stated earlier, there is a need for closed systems. It can start small and grow. GMP manufacturing utilizing the same connectors can be taught, and the scalability of these closed system connectors from small volumes, from your flask, from your lab, to large batch manufacturing. Single-use aseptic connectors can be used from seed train, expansion, inoculation, filtration, concentration, and to your final fill. Let CPC and aseptic connectors grow with their process. Work with your suppliers, channel partners, to think ahead when designing single-use closed process to reduce the challenge ahead. Considerations need to be taken when you're thinking about a closed system. There are several options out there when you're putting your process together. There are a couple ways to achieve what you want to think ahead. Does your process utilize different size tubings, different material of tubing? Let's say you need to use a tubing for a pump, and then you want to flow into your final fill process, for example. Are you limited to the type of tubing due to the process you're using? Does your process require other accessories to achieve your connectivity need? Is there calibration and maintenance that need to be kept up? How much clean room floor space do you have in your suites? Do you have room to move your equipment? Have you tested the process for only having drug product and buffer on your final product? Carefully evaluate your needs and work with the suppliers when you design your process. Utilizing some of the history that biopharm has gone through can be beneficial to leverage to avoid any problems in the future. Therefore, aseptic connectors are what you need to look into. Choose a supplier that's flexible. You will find that choosing CPC aseptic connectors will allow for that flexibility, easy to use, repeatable, sterile, transfer of fluid in any environment. And the reason you have to look at aseptic quicks as a closed system connector is it has been well validated, robust, and simple to use. It's single use, reduces cleaning and sanitation. It's a sterile connector that can be used in a non-sterile environment. The simple 3-step process is easy with the flip, click, pull operation. It's genderless and it simplifies the system setup, ordering, supply chain, operator training, inventory. And it connects to different types of tubing. And it doesn't matter of size or material. No extra equipment is needed. And with the variety of sizes that the connectors come with, you can find a size that would fit your needs. Before I end here, I'd like to leave you with several questions. Who are the partners that you value? Can suppliers' products grow with you and scale up with you or scale out with you? Will your suppliers help every step of the way in your connectivity needs? How do the products and components help you with the ease of use? Do they help train your operators? How do companies and products support your manufacturing needs? And are they flexible and agile? Have you looked at the overall cost, including the cost for maintenance and training? Do your supply team understand cell and gene? CPC can do all this with our portfolios of connectors, products, and disconnects. I'll also be looking forward to introduce an exciting new product from CPC that in the coming months to optimize your therapy process. With that, I will end. If you need more information, please look us up at www.cpcworldwide.com at CGT. You're also more than welcome to come to our booth at Meeting on the Med. Thank you.